Hello everyone, welcome to Assist Biology. In this video, we are going to discuss about vulva formation in C. elegans, and this is the second part from the topic. This topic is from the unit of developmental biology, and this will be helpful for B.Sc. M.Sc student, CSIR NET, DBT JRF, ICMR JRF, ICAR NET, and GET Life Science aspirants. So we have already discussed this diagram in our first video. So you can see here the position of gonad, that is represented in the black color, the position of anchor cell in the red, and you can see the 6 BPC, they are spherical in shape outline. Then this is the only the enlarged part of this first one. So you can see the gonads, the anchor cell, and the BPCs, that is the vulval precursor cells. So we will recapitulate what we have learned in the first video. So the steps are as follow: the generation of vulval precursor cell BPCs. As you know, these are the initiator of vulval development. So they will be formed first. Then the BPCs are actually 6 cell from the 11 cell of the ventral epidermis, and they are specified, that is they are quite distinct from the other cells of the ventral epidermis. Total 11 cell in the ventral epidermis, and from them 6 are BPCs. Then the vulval precursor patterning. Although there are 6 BPCs, but their degree, that is the vulval precursor patterning, is different. So signal from the gonad and the signaling among themselves, that is the intercellular signaling between the all BPCs, determine their fate. So there are 3 type of vulval lineages, that is 1 degree primary, 2 degree secondary, and 3 degree tertiary. So 1 degree vulval precursor cell are the cell which are receiving the highest concentration of the signal, that we will discuss. Secondary or 2 degree are receiving little bit lesser concentration, and tertiary or 3rd degree cells are not receiving any signal. So generation of adult cell, the adult vulva is having 22 cells, as you know. So here you can see the diagram. The diagram is one of the most important. So here you can see the gonads, that is black in color, the red colored anchor cell. So the anchor cell secretes lean 23 protein. Lean 3 protein. Lean 3 protein is a epidermal growth factor. So epidermal growth factor, it is a signal, or you can say this is a ligand. So the cell which is receiving the highest concentration of the lean 3 ligand, that is the P6P. You can see here the P6P is the nearest to the anchor cell, so it is receiving the highest concentration of the ligand, that is the lean 3 protein epidermal growth factor. And the other 2 cells, that is P5P and P7P, which are adjacent to the P6P, they are receiving the lesser concentration of the ligand or signal molecule, that is the epidermal growth factor or lean 3 protein, you can say. So the cell which are receiving the highest concentration will become the 1 degree or primary VPC, or the other 2 cells which are receiving the lesser concentration will be the secondary VPC, or you can say 2 degree VPC. And the primary VPC, that is the 1 degree VPC, will form 8 cell, and the other 2 BPC will form 7 cell each. So there are total 22 vulval cell. And here you can see the P3P and P4P, that is the 2 cells that are the distant cell from the anchor cell, and P8P, these 3 cells are most distant from the anchor cell, and they are not receiving any lean 3 signal. That's why they will become the hypodermal cell, and they are called as tertiary vulval patterning lineage, or they are 3 degree vulval cell, and they will become the normal hypodermal cell. Then what are the steps? We have already discussed in the diagram. Let us learn the statement wise. Okay, so first of all, vulva is present mid-ventrally. As you know, the position is the middle of the body, and it is present in the ventral surface of the body. So the first vulval precursor cell will be formed, because as you know, these are the initiator of the vulval development, so they must be formed first for the initiation of the process. Then there are 6 epidermal cell, that is the P3P, P4P, P5P, P6P, P7P, P8P, and they will be called as the vulval precursor cell. And the VPC or vulval precursor cell, they are called as equivalence group, because they have equal potential to form the vulva. So according to the different concentration of the ligand or signal they are receiving, they might be become other cell, means the fate will vary, but as they are having same potential, they are called as equivalence group. Now the anchor cell secrete the lean 3 protein. Okay, always remember the anchor cell secrete the lean 3 protein, and which is epidermal growth factor. So the VPCs, that is the vulval precursor cell which is receiving the highest concentration of the signal, that is the lean 3 or the epidermal growth factor, will become the 1 degree VPC or the primary VPC. So as you have seen in the diagram, P6P is the closest, so that will receive the highest concentration of the that lin3 protein or the epidermal growth factor, so they will become the primary VPC or 1 degree VPC. And the adjacent to the cell, that is the P5P and P7P, will receive the lesser concentration of the lin3 protein and they will become the secondary VPC. So primary and secondary VPC is according to the level or the concentration of the signal, that is the ligand molecule lin3 protein or the epidermal growth factor they are receiving. And as there is a ligand, there must be a receptor for the ligand. So lin23 receptor act as the receptor for the lin3 protein. So this will be receptor. So LAT23 is the receptor and lin3 is the ligand molecule. So lin3 is the ligand and the LAT23 is the receptor. So there must be a ligand for each receptor. So LAT23 is the receptor and lin3 is the ligand protein. So what happens? The P3P, P4P and P8P did not receive any lin3 protein signal and they will become the hypodermal cell or they will form the tertiary vulvar lineage. Then the 1 degree VPC inhibit the 2 degree VPC, that is the primary VPC inhibit the secondary VPC by secreting lin12 protein. So lin12 protein cause the lateral inhibition. So here you can see, here in the diagram you can see here the lin12, these are the lin12. This lin12 is secreted by the primary VPC and it will cause the inhibition of the secondary VPC to become the primary VPC. So the primary VPC inhibit the other secondary VPC, that is the P5P and P7P, to become the 1 degree VPC. That's why they can't become the 1 degree VPC and they remain as the 2 degree VPC. And lin39 protein is essential for the specification of VPC, that is which VPC will be what, that will be specified on the basis of lin39 protein. And the LAT23 is a receptor tyrosine kinase. There are number of receptor tyrosine kinase and this follows a MAP kinase pathway. So receptor tyrosine kinase means this will lead to the phosphorylation of the tyrosine protein residue on several protein molecule, tyrosine amino acid residue or on several protein molecules. Now you can see here lin3 is the ligand molecule and LAT23 is the receptor. You must remember lin3 is the ligand and LAT23 is the receptor molecule. That means lin3 will be secreted by the anchor cell and LAT23 receptor will be present on the vulval precursor cell. So LAT23 will form the primary VPCs, that is the receptor, and the ligand interaction will form the P6P, will turn the P6P into the primary VPC, and this primary VPC will secrete the lin12 protein. Okay, lin12 protein will inhibit the other two VPCs to become primary VPC. So due to action of the lin12 molecule, they can't become the primary VPC. So DSL activates in the primary VPC, that is DSL protein will activate in the 1 degree VPC, and the DSL will leads to production of the lin12 molecule. So lin12 molecule prevent the secondary VPC to become primary VPC. So as the P6P on the both side of the P6P or on the lateral side of the P6P, P5P and P7P are lying, that's why this process is called lateral inhibition. At the cell, that is the P6P, that is the 1 degree VPC, is laterally causing the inhibition process of the P5P and P7P. So this is called as lateral inhibition, and this lateral inhibition from the secondary VPC, that means they will prevent the VPC to become primary VPC. And here what happens, how it is causing the inhibition? So what happens, the lin12 molecule, that is lin12 molecule, cause the inactivation of the LAT23 receptor molecule present on the VPC, that is on the secondary VPC. So as the LAT23 receptor are being inhibited by the action of the lin12 protein molecule, so what happens, they will not become able to receive more and more lin3 protein signal. So as the receptor number is decreasing, that is the LAT23 receptor number is decreasing on the VPCs, they will not be able to they will not be able to show their response towards the signal, that is the lin3 molecule, and they will become the secondary VPC. So mutation cases are always most important. Okay, so mutation cases, so always in examination question come from the mutation case. So you must know you must know the reason and you must remember the cases. So first of all, low lin3 or LAT23 molecule, that is the lin3 is the ligand and the LAT23 is the receptor. So no primary VPC will be formed, because as we know the anchor cell must secrete the lin3 ligand and the VPC will must receive through the LAT23 receptor, as there are low in the quantity, that means they are present in low quantity, so the no primary VPC will be formed. Then if anchor cell are destroyed, so as you know, the anchor cell is means the, as the name suggests, they are anchoring the whole process of the vulval development. So as the anchor cell will be destroyed, there will be no Lin3 molecule, so all VPC will become epidermal and no vulva formation will take place. Then if Lin3 is blocked or mutated, so Lin3 is the ligand molecule that is responsible for the formation of the 1 degree and 2 degree VPC, so no vulva formation will take place. So if gonads are destroyed, this is one of the most important, you must remember, fourth one. So if gonads are destroyed, vulva formation will be there. Vulva formation will be usual process. Usually the vulva will be formed without any disturbance, because the anchor cell is secreting the Lin3 molecule, that is the epidermal growth factor protein molecule. Okay, so if gonads are destroyed, there will be no any abnormality in the vulva development. Then if 3 degree VPC are destroyed, as you know, 3 degree VPC or tertiary VPC don't have any role in the vulva formation, because they are becoming hypodermal cell, so vulva will be formed. And if anchor cell position is changed, means the position of the anchor cell is changing, then the vulva formation position will be changed, that is the position of vulva will be changed, because as you know, all the VPCs, that is the vulval precursor cell, are equivalence group, that means they have equal potential. So as usual, when they will receive the signal from the anchor cell, that is the Lin3 protein molecule, then they will become the vulval, that is the primary vulval lineage and secondary vulval lineage. And if the all three central VPCs are lost, that is the your P5P, P6P and P7P are lost, then the rest 3 VPCs, that is a P3, P4 and P8P will form the vulva. So mutation cases are always important, you must remember this. Out of this, the fourth one is important, very important, and the 7th one is very important. So 4th and 7th, these are important. This is the end of the video. Thank you for watching, and if you are new to this channel, please subscribe the channel and press the bell icon for getting more updates.